thank you. If our four speakers would stay on, and if anyone has a question that they would like to ask them, please feel free to do so now. Okay, here's one question, Dr. El-Traj. Have any of the technologies like mRNA been used before in animal health, or is this completely new?
So for mRNA, nothing has been. There's been research and development on mRNA in animals. There's been like mechanisms that are similar to mRNA, which are called self-replicating RNAs, that actually Harris vaccine started and are utilizing it in a replicon fashion. Now by Merck, which has been utilized both in swine as well as poultry industry, and have been very successful in preventing diseases in both those species. So yes, in that form and that fashion of self-replicating RNAs, yes, it has been utilized. Now the delivery of what I call naked RNA or naked DNA has been very limited. I think there's been a couple of DNA plasmids that have been utilized for avian influenza within the poultry industry and utilized for stockpile purposes, but for commercial use, it's very limited. Again, with COVID-19 coming to the forefront, and with cost of these materials coming down, it becomes much more of a reachable goal to utilize these technologies in the near.
Okay, thank you, John. This question is for Dr. Casaubon. Question is, can this type of technology, the SIR, work for other diseases like ILT or bronchitis?
Oh, yes, definitely. I think that it can be applied to any type of contagious disease example. And yeah, definitely, that's also doable for IB and ILT. And be sure that we are currently assessing some of other issues as well in using the same system. So yes.
Okay. Next question is for Dr. Linares. Question is, what's your opinion of using more than 2 to 3 IB vaccine strains in one application?
So what we need to consider is that whatever we do, it has to provide relevant protection. And the rule of thumb is to do the simplest program that will provide relevant protection. So we've been able to execute with just 2 serotypes. As a matter of fact, the more we look at vaccine detection and combine it with the effectiveness of the program, when we use 1 or 2 vaccines, we have great vaccine take in the hatchery, excellent protection, and we basically would not add serotypes unless we really needed to have relevant protection.
Okay. Dr. Sesti, this question's for you. It says, how would you compare the ILT challenge level between Purdue and the Delmarva Peninsula, for example, or between Argentina and Delmarva?
Well, if you look at Argentina, Argentina has actually 2 main regions of broiler production. One of them is it's right outside the capital, Buenos Aires, and the second one is about 200 kilometers at northeast part of the country. This second one is really a very high density area. I mean, you have a lot of small, medium producers side by side, layers, broilers, breeders, etc. So not talking about volume, but I'd say this area in Argentina is pretty much a, let's say, a small Delmarva region, very similar. A lot of companies side by side, and the dissemination and viral dissemination in that region is really complicated. Yeah, it's similar. Peru is totally different. The companies are you don't really have very high density areas, so it's a little better in that sense.
Thank you. This question for Dr. Casaubon. how do maternal antibodies affect the results of the SIR trials, and was this taken into account in the trials you showed today?
Okay, so I'd like to remind that we used actually commercial broilers. They were provided with medium level of maternal antibodies, about 5.5 log2, so that's not very high, but there were definitely maternal antibodies to these broilers, and the vaccination was done in front of these maternal antibodies. So, and I'd like just to remind everyone that since it's using the Marek technology, which is cell-associated, so that's today the best technology which helps to elicit a proper immune response and vaccine immunity development in the face of maternal antibodies. So that's why we were willing to run this study in commercial birds instead of SPFs.
Okay. This question is for Dr. Elatrache. How long before we will begin to see these technologies like LNPs and mRNA commonly used in poultry production?
Wow. So yeah, great question. Delivery of whether it's RNA, whether it's self-replicating RNA or DNA. I would say in the very short near future. I would say USDA has already allowed this by releasing the 800.213 memo. They already have regulations in place, so U.S. will be the first spot where we'll see this. I think you'll see this introduced into more or less the larger livestock, whether it's swine or cattle, and then I think it'll trickle down into poultry. So bringing a nice crystal ball to structure here and saying, okay, how soon will it be? I think it'll be probably within 2 to 3 years. I think realistically, you'll have that advent coming into the poultry industry. And I think you'll be able to take advantage of the LNPs, the structural protein engineering, as well as delivery of these nucleic acids that will enhance the host immune system, whether it's poultry, swine, cattle, will enhance the host immune system to both arms of the immune system, whether it's cellular immunity or whether it's immunology.
Thanks. And this question for Dr. Linares. How does the application method affect response to the IB vaccine that's used? For example, spray versus gel.
All right. So for example, when we obtain our label claims for IBRON, we tested the efficacy of the vaccine both by spray and gel, and which resulted in equally effective results. And in the field, we have executed with both spray and gel. The important thing is that the equipment has to deliver a uniform pattern of spray or gel droplets into that cheek box. And basically, you do that visually. You could do that with paper. You can do that with plexiglass calibrated plate. But the fact is that you want to deliver the vaccine uniformly to those 100 chicks in the box. And then, of course, we had the capacity to do vaccine detection. And we have seen good vaccine delivery and vaccine take, both with spray and gel. But there's so many variables, it's just, but the fact is that the equipment has to do that on a consistent basis and then to vaccine detection.
Thank you. And this will be our final question. This question is for you, Dr. Elatrache. Regarding the design of next generation coronavirus vaccines, isn't the focus on a hypervariable or glycosylated spike a bit short-sighted in chasing a moving target? Are there other conserved proteins that could be incorporated to provide a synergistic, cross-protective, and more durable vaccine design?
Oh, undoubtedly, yes. The answer to that is yes. I don't think the spike is short-sighted. I think it's always the key immunogenic protein is, and I think what you're seeing. Let's take mammalian species versus poultry species. You see much more drift in mammalian species from the immunological response from the mammalian species than you do poultry species, where there really is an issue with drift in the human species. What you've seen today is you see an advent of variants already popping up with COVID-19. And these variants are in the receptor binding domain. But what you've seen also is that the vaccines that have been developed against the original strain holding up to these mutations. So I think we're opening up a different realm here of the ability for these structured proteins that are being delivered by vector systems to provide an optimal protection. Now, if we talk specifically for bronchitis within the poultry industry, it's a different quagmire because we've been utilizing live vaccines for the longest time. And we've been changing live vaccine programs. And I think what's settling down now is that with the use of one or two, two strains of bronchitis, we're able to calm down the spread of other serotypes or other genomes throughout the globe, whether it's in the U.S. or whether that's throughout the world. So the effect of really utilizing a minimal amount of bronchitis vaccines to protect across different serotypes and genotypes is working. It doesn't mean that we're going to get new ones popping up constantly. We will, but I think you still can resolve by spike protein. And yes, again, if you can drive an immune response for something more conserved, such as nucleocapsid or something else, yes, it's advantageous. And I think with the new vectors, last comment on this one, I think with the new vectors that are non-competing, you can do that. And you can do that effectively. So yeah, inclusion of that, but I think also there's going to be a lot of improvement that you see with specific gene of interest, such as spikes that are the major immunogenic genes of interest and translated into protein.
Thank you, Dr. Eltraj. And that's going to wrap it up today. We're completely out of time. So thanks again to everyone for attending. Thanks to our speakers for your preparation. Everyone have a great rest of your Tuesday. Thanks, everybody.